Alright, I'm going to talk briefly about this paper where they resurrect a clinical antibody. Basically what they did is they had some old archived cells which were producing some antibody, and these cells were too old to grow up as a normal hybridoma and produce a large quantity of that antibody. So what they did was they used mass spectrometry to determine the variable sequence of the antibody, so the part of the antibody that binds to proteins, and once they determined that, they then they made a whole new sequence, put that in a plasma, and had some other cells express the antibody. They were able to produce a bunch of it and show that the antibody is still bound to its original target. Another thing I'd like to point out is this was made between a collaboration between the University of California, San Diego and Genentech. Genentech is an old biotech company that's been around for a long time. I'd say about around the 1970s. They were one of the original companies that made insulin, which is one of the big first biotech accomplishments. And they also produced Herceptin, which is an antibody that binds the molecule HER2, which is overexpressed in certain types of breast cancers. It's one of the few cancer treatments that's been successful and profitable. So a few more things about Genentech is Forbes magazine and some other magazines have listed them as a great place to work. I think one year they were like #2 to Google as one of the best companies to work for. So Genentech sounds like a nice, interesting company. And this paper is fairly recent. It was in 2011. So why is this research here significant? One of the main reasons, well, I mean, there could be a lot of unforeseen things that are good about it, but one potential use I see for it is you could take the information from any immune person's immune system and transfer that to someone that's not immune. So they actually don't mention that application in this paper, but theoretically, you could take someone who is resistant to AIDS. For example, there's these people that are classified as HEPs, which is highly exposed, persistently seronegative. And these HEPs people are in contact with AIDS all the time. Often they are prostitutes or commercial sex workers, and they never actually get AIDS. So there's different theories about why they don't get AIDS, but one possible theory is that they may have neutralizing antibodies to the virus. Now, using this method of just getting the antibody protein and using mass spec to figure out the variable region and then producing more of the antibody, using this method you could get antibodies from those HEPs people, then mass produce antibodies resistant to the AIDS virus, and then transfer that to other people, making them resistant as well. That's just one possible application I'm throwing out there. Here in this paper they focus on lymphotoxin protein, lymphotoxin alpha, which is similar to tumor necrosis factor, and they claim that possibly if you could block lymphotoxin alpha, which is on the surface of some cells, then you may be able to help with the arthritis disease, where there's always a lot of inflammation. And so they have this old archived cell line, these mouse ascites. I'm not sure what an ascite is, but they have these mouse ascite cells, and they get the antibody from them, and then they use this mass spec method to reconstruct the antibody. So I'm going to go through here, just kind of skim through some of the paper, maybe pick out a few things to talk about. Try not to make this video too long. I know it's quite technical and maybe not interesting to everyone. Here's their general process of how of what they do in this paper. They purify the antibody from the mouse ascites, then they do mass spec analysis. So when they do their mass spec analysis, what they do is they cleave the antibody into many different fragments and then do mass spec. But what they do is they have different samples where they cleave the antibody with a different protein. So they actually use 6 different proteolytic enzymes in 6 different samples to chop up their protein. And so they'll end up with a bunch of different fragments, but the fragments from different samples will overlap. In some ways, this kind of reminds me of the shotgun sequencing that some groups did when they tried to sequence the human genome. So once they determine the sequences, and they actually align these sequences with the known V, D, and J segments that could be possible in a mouse antibody. And then what they do is they match up that antibody with an antibody that they already have the sequence for that's very similar. I guess they have a whole archive of sequences for different antibodies. So then they designed oligos to mutate that DNA sequence to this antibody that binds to the lymphotoxin alpha that they're working with in this paper. And then what they do is they make these vectors, and then they transform that into cells and have them express the protein. They purify their protein, their antibody against the lymphotoxin alpha, and then they see if the antibody binds to the original target, the lymphotoxin alpha, the re-engineered, reproduced antibody, and they show that it actually can do this. So I'm going to zoom back out here, start scrolling through the paper again. So here, let's look at some of the results here. So here in this figure, they're just showing that the original antibody from the mouse ascites really does bind lymphotoxin alpha. So this is an ELISA graph, and they're showing as they increase the antibody concentration, they don't allow the antibody binds to lymphotoxin alpha, and it's not allowed to bind to the target on the plate. If it did bind to the target on the plate, it would have a high OD. This is kind of different than straightforward ELISA. So here's kind of a little drawing of it here. They have TNFR2 Fc coated on the ELISA well plate surface, which binds the lymphotoxin alpha. Then, pre-mix, before they put anything on the plate, they pre-mix lymphotoxin alpha with these mouse ascites or with this other compound that's supposed to bind lymphotoxin alpha. And so when they premix it and bind it onto the plate, if it's already bound to this protein, it's not going to bind to the coated TNFR2C. But if it is bound, it won't bind to that, and you won't detect anything. So as you can see, as you increase the concentration of the antibody or the antibody with these ascites, you detect decreased absorbance on the plate. I'm sure that's a little bit confusing, but basically they're just showing that the ascites do bind the original protein. Here's some FACS data, some fluorescence activated cell sorting. It's just another way to show that these ascites do bind to the lymphotoxin alpha. So here's the peak for the ascites. On the x-axis you have the intensity here. Oh, actually, over here they have mean intensity. Over here they have the lymphotoxin alpha expression. And they show the control, this just isotype, doesn't bind it very much. So that's that. So that was just the initial work to show that these mouse ascites are worth working with. Let's see. Okay, here they show some of the results they got as they were trying to do the mouse spec. So here they show when they tried to align their sequencing results from the mass spec with the original, with the known sequences, there was some differences. So all the things in red were mismatches. So this line is the predicted heavy chain variable region of the antibody, and up here you have actually the correct sequence they obtained from the mass spec. So there are some differences here, and then this line is just continuing from above. And as you know, like with an antibody, you can have the an antibody has this Y shape. It has two chains. The heavy chain is on the inside, variable chain on the outside, and the part of the antibody that binds to the protein is on this tip here. So the variable part is just on these tips here. And the light chain and heavy chain are connected here just by a disulfide bond. But so anyway, they're just showing the sequences they got from the mass spec. I won't spend too much time on that. And here on this page, they're just going through some of the mutations they had to make to convert their existing antibody into the antibody that would bind the lymphotoxin protein. I'll move along here. Okay. So here they're comparing the original ascite antibody with the newly produced chimera antibody with the compound that just binds TNF, or the lymphotoxin alpha normally, along with a negative control. So this ELISA is different than the ELISA before. This one's more direct. They just coat the plate with lymphotoxin alpha, and then above that they put one of these four things right here. So they'll put like the ascite or the chimera antibody. And so these high binding things are the original ascites, and then also their re-engineered, reproduced antibody. They both bind the lymphotoxin very well, so that's great. Then they just show the same thing with some fax data. And here you see that the negative control doesn't bind very well, but then the others do. And then here they show that with some other cells, these HUVEC cells, which are expressing lymphotoxin alpha, they show that if their compounds bind to the lymphotoxin alpha, these cells won't be able to produce ICAM, like normally ICAM would be produced if the lymphotoxin alpha is produced. So they're just kind of showing here that this could possibly have some clinical applications. And then this figure here is just some SPR data, some surface plasmon resonance data, to figure out exactly how tightly their re-engineered antibody binds to lymphotoxin alpha. So they were able to obtain some good dissociation constants. So they have the different titrate. Each one of these lines is a different titration that they use, and then they have their on rate and their off rate, and they're able to calculate the KD. So yeah, basically, I mean, they're just showing that that this process does work. Here, I just want to mention here, like, some of the potential uses for monoclonal antibodies. I mean, you can try to use it to treat cancer, inflammation, autoimmunity, and infectious disease. There's a lot of potential applications. Let's see if there's anything else I want to mention before wrapping up here. Oh, here, this process, or the program they used as they were sorting out their bioinformatic data from their mass spec was GenoMS. They did say they had to make some modifications to it in order to get everything to work. I think that's about it. So they just end off by saying that this accomplishment opens up the possibility of resurrecting other antibodies from obscure sources. And then I don't think they necessarily have to be obscure. I mean, just any time you find some animal or human that's immune to something and you want to be able to figure out the sequence of the antibody directly, like instead of from the DNA or RNA from the B cells that are producing the antibody or something, like if you just have the antibody straight from the blood, just that protein, you can then use mass spec to figure out the sequence, and then you can, once you have the sequence, you have it in the format of information. You can transfer that information into any other thing. You can make a lot of that antibody and potentially treat many illnesses and diseases. So yeah, that was a pretty interesting paper. Thanks.